Good morning, everyone. Welcome to the NanoBio4Can Webinar Series. First, I'd like to give a brief information about the project. NanoBio4Can project, a mobility scholarship program for postdoctoral researchers, aims to develop nanobiotechnologies for innovative therapeutic approaches in cancer. While SUNUM is the coordinator of the project, İzmir Biomedicine and Genome Center, Koç University Research Center for Translational Medicine, and TÜBİTAK MAM are the implementing organizations. For detailed information, please visit nanobio4can.net. Today is our fourth webinar. We have Dr. Saif Ullah Afridi from TÜBİTAK MAM, Dr. Senthilkumar Palanisamy and Dr. Shoeb Anwar Ansari from SUNUM. Each researcher will speak for 15 minutes, followed by a 5-minute Q&A session. We will organize this monthly webinar within the framework of the MSCA COFUND Grant Agreement dissemination and communication obligations. This webinar will be recorded. It may be posted on YouTube. If you don't consent, you may keep your camera off. Dear all, welcome once again to the webinar. Thank you again for your valuable contributions. We will start with Dr. Saif Ullah Afridi. The stage is yours. Would you introduce yourself to us while starting the webinar?
Thank you very much. Hello, everyone. My name is Saif Ullah Afridi, currently working as NanoBio4Can MSCA COFUND Fellow at TÜBİTAK MAM Research Center. Prior to continue my presentation, I will mainly focus on these 5 key points. Personal biography, project detail, which will be further subdivided into background, focus point, aim and objective, strategy, result, conclusion. Dissemination activity, acknowledgement, and question and answer. So, as I mentioned, my name is Dr. Saif Ullah Afridi. My academic and research was started almost 2 decades before, after graduating from one of the famous institute, the Dr. Panjwani Center for Molecular Medicine and Drug Research, University of Karachi in 2015 with a scholarship MPhil PhD study. This is the Dr. Panjwani Center from where I graduated in 2015 with a fully funded scholarship. After graduated, then I joined Kamset University, Islamabad, followed by University of Lahore, Islamabad for 2 years, working as assistant professor. And then I was able to find a fully funded postdoctoral research opportunity by this postdoctoral training opportunity at Hangzhou Women and Children Medical Center at Institute Pasteur of Shanghai, Chinese Academy of Sciences, with a good research fund of almost 75,000 USD and salary with a good salaries. After completing my postdoctoral research training from 2018 to 2021, I returned back to Pakistan and served another university, Kuwait University of Science and Technology, followed by another university, Sarhad University of Science and Information Technology, Mardan campus, as an associate professor. During this tenure, I secured around about 5 research projects, completed 4 research projects, 3 from China, 1 from my home country, and the 5th I didn't join. That was went from the Brazilian Biotechnology Research Center, but meanwhile I was able to find the NanoBio4Can postdoctoral opportunity, so I skipped the Brazilian project. So I completed 4 research projects as a PI, and these are the projects which I went and completed as a principal investigator. And these are the reports which I already submitted and completed the project successfully. And overall, this is my honor and awards. Like I won Early Career Boot Camp Project Presentation Award from Germany in 2021, and I presented my idea in front of international jewelries. Similarly, I went OCPC International Young Scientist Award from China Outstanding Young Scientist Award, Project Presentation Award from the Women and Children Medical Center. These are, you can see my names, and these are some awards which I won and completed. Similarly, joining my first university, Kamset University, Islamabad, which is almost ranked right second in my country. I won the Research Productivity Award for the year 2017. And these are till now working as an assistant professor and postdoctoral researcher. Till now I have 29 publications and through 3 book chapters. With the ranking of my publication is 83% is ranging as Q1 and Q2, in which 54% is Q1 and Q2 is 29%. With an H index 16 and I index 19, with an impact factor of more than 150. And these are the research grant which I went deliver awards, organized some conferences, nominated for different research project award. And meanwhile, I taught different courses to MS and BA student at this university, which I already mentioned. Almost 20 different courses I taught to different students, MPhil, master and BA students. Relevant to this current project, we have already published good research articles from my previous postdoctoral training, including cell discovery, International Journal of Cancer, almost all were Q1, and some from my own project, like this one from my own project, and this is also from my own projects. So we published good research article from the current, from the relevant background. So grounding on this background, my current project is focused to dissect B7 associated immune landscape and tumor immune microenvironment, recapitulation and exploration. As we know, cancer rank is a leading cause of death and an important barrier increasing life expectancy in every country of the world. According to estimate from the World Health Organization in 2019, cancer is the first or second leading cause of death before the age of 70 and ranked third to fourth in 23 countries. Lung cancer is also remained the leading cause of cancer death with an estimate 1.8 million deaths, followed by colorectal cancer, liver cancer, stomach and female breast cancer. The global cancer burden is expected to be 28.4 million cases in 20 by 40, a 47% rise from the 20 by 20. So here, the research problem is, despite numerous strategies to explore, understand the interaction between the tumor and their environment, microenvironment remain unexplored because people are trying but still is unable to cure cancer. Sometimes like immune cell are facilitate cancer progression instead to suppress cancer. For instance, tumor-associated macrophages sometimes facilitate cancer initiation and progression as well as metastasize and help in angiogenesis and suppress the cytotoxic T cell to attack cancer. So to explore this opportunity, there are current treatment for cancer which is available is chemotherapy, radiation therapy, chemoradiation therapy, gene therapy, and immune checkpoint inhibitor therapy. But immune therapy is considered to be the gold standard method in order due to low cytotoxicity and high specificity. The immune therapy, currently there are 4 famous adaptive immune therapy in which the patient T cell are removed, engineered, and reinfused to kill the cancer cells. Similarly, the macrophages are loaded with the APC are loaded with the tumor antigen and they reinfused to the patient and try to eliminate the tumors. The other strategy is to cytokine-based therapy in which the anti-cancer or T cell activating cytokine are injected to the patient, but they have highly cytotoxicity. So the most important is the immune checkpoint inhibitor therapy are still considered to be the gold standard, where the specific immune checkpoint is blocked by using antibody or some small molecule to target the cancer cell. But still there is a gap to overcome this challenge because we need to explore more immune checkpoint to completely eradicate cancer. To overcome this issue, we propose this area of target, which is relevant to B7 family immune checkpoint, which need to be explored very well in order to understand the scenario or the crosstalk between the tumor cell and T cell. So the aim and objective of my project is to dissect tumor immune microenvironment at cellular and molecular level by targeting B7 family related interaction. And the objective of my project is to identify unique cell marker, cytokine, enzyme, or pathogen using advanced dissecting approaches to evaluate the potential role of the candidate marker in tumor immune microenvironment regulation analysis, to validate the result by using the respective cell line, stem cell, organoid, or real clinical sample, and to select the highly active new marker for specific pathway analysis using the standard reporter assay protocol, and to study the new marker in multi-center cohort samples. This is the roadmap. In first year, we are supposed to explore the new marker, and this second or the last year, we are supposed to validate this marker in real samples. This is the detailed procedure which we already apply in optimize the marker. Like after collecting the sample, we design the panel, we stand chromogenic base and fluorogenic base staining for the selected marker, and then we proceeded for B7 marker TSA and TMA optimization. And currently we are in monochromic and polychromic image stage. This is the general workflow, how we are dealing with this. After the patient diagnosed with the cancer, lung cancer, the minimum criteria is the tumor should be at least more than 2 centimeters. For instance, this is a real sample of patient which have 2.16 centimeter tumor and lungs. So we are supposed to collect both bloods and tissue of the cancer. The cancer, again, 3 types: tumor, peri-tumor, and peripheral normal tissue. And then we are supposed to proceed for unique cell marker analysis, and then to see its correlation or life expectancy for further analysis. But before doing this, we need to optimize the protocol, and we were supposed to optimize our protocol by using standard operating procedure. For instance, we optimize 5 parameters dealing with this staining procedure, like antibody concentration was optimized, opal dye concentration was optimized. For each marker, antigen retrieval buffer was very important. We optimized that condition as well. Antigen retrieval buffer pH was also considered as a key point for staining the procedure, and antigen retrieval condition. So these are the parameters which we already optimized, and hopefully we will proceed for further. And first, we are supposed to check individual markers, individual stainings, and DAB staining, non-chromogenic base staining. And after validation, then we were supposed to proceed for fluorogenic base approach. So these are different markers which we optimized and tested. And then we are proceeded for multispectral immunohistochemistry staining, and to see the different marker correlation and co-expression in different cancer, like ovarian cancer, lung cancer, hepatocellular carcinoma patient cancer. And this is the B7 panel, which we already optimized previously with other tissue, and now currently we are supposed to optimize the same panel with lung cancer patients. And after the optimization, you're supposed to see the multispectral immunohistochemistry and a cohort-based assay slide, which we call them TMA, tumor microarray, which contain more than 100 patients per slides. So in conclusion, we successfully optimized and validated all B7 panel antibody using DAB and TSA single staining protocol. Hopefully, we will be able to explore the desired new B7 family associated marker and regulatory pathways. We hope that our finding will help in target-oriented pharmacophore design, screening, and analysis. We expect that the unique B7 associated marker regulatory pathway will open new window for researchers and healthcare professionals to manage and treat cancer patients. The desired finding will be shared with the scientific community. And these are the references on which my current project is grounded. And these are the dissemination activity that I participated in almost every training subject to availability, including offered by my grant to TÜBİTAK Research Center, SUNUM, Cross Center, Izmir, and Koç University, as well as we also created linkage with different hospitals, like this is Sakarya University Hospital. From here, we are getting our samples. Already, we collaborate with the Gebze Technical University for student guidance and supervision and trainings, and also participated to increase our upgradation. For instance, participated in the Nobel Laureate Seminar, 2022 Nobel Laureate Seminar. And to link with industry, we already participate and connected with the industry, like we visited Trust Life. To motivate youths, we delivered public speaking and presented our areas in presentation to public school guide so far. But before this, we can see that in this 1 year, whenever I completed so many certificates, reviewed some research article, work as a reviewer for different journals, completed different online seminars, completed some courses offered by Coursera, and almost I got more than 20 certificates. And finally, I would like to acknowledge SUNUM and NanoBio4Can for giving me this opportunity to present my work in front of my colleague, researcher, and public. I would also thanks to my supervisor, Professor Abdullah Karadak, and my research team. And I would also like to thank my co-supervisor, Professor Devram from Koç University. And I would like to thanks to Sakarya University Training and Research Hospital for providing the clinical sample, real clinical sample. It's also, I would like to thanks my previous postdoctoral research groups, with the help of which I was able to optimize the skip parameter previously, and now I'm applying the same procedure and protocol for dissecting lung cancer here at NanoBio4Can and TÜBİTAK. I would be happy to answer if you have any question. Thank you.
Thank you, Dr. Saif. Q&A session is open. You can just speak up, or you can use chat box. We have a question on chat box, Dr. Saif.
Yeah.
Müge Özkan wrote. I can read her question. Saif, thank you for the presentation. Maybe I missed it, but the main idea is to identify a lung-specific tumor marker. So for that, you need to have a source data of candidates, then try on protein level. Where your source data is coming from?
I saw. Thank you, Muge, for your question. Regarding source data, as I mentioned previously, I have already worked with a good research team at Institute Pasteur of Shanghai Chinese Academy of Sciences, Nandowamin Institute and Medical Center. But currently, we have good resource data from the Sakarya University Hospital, from where we get clinical sample, Medifol University and Koç University Hospital as well. We got real sample from that. While regarding marker base, so I have a strong collaboration with my previous postdoc training groups, and they have vast experience and different marker crosstalk and correlation. So we are currently just validating the same approaches here within the limited time frame. I hope you pick my answer.
Do you have any question? Ana Luisa asked a question. Thank you for the presentation. Why did you choose as inclusion criteria tumors larger than 2 centimeters?
Yeah, thank you again. This is a standard operating procedure. Like most of the less than 2 centimeter have limitation by tissue processing and washing, then optimization, removing, as well as the standard criteria from our previously optimized protocols. So we are just following the same optimized protocol in order to overcome the time constraint and to complete our project within the times.
If you don't questions, we can go through the webinar with Senthil. Thank you, Dr. Saif, for your presentation.
You're welcome. So should I stop my sharing?
Yes, please.
Okay.
Now we have Dr. Senthilkumar Palanisamy. Stage is yours.
Excuse me. Is it okay? Yeah. Yeah. Good morning, everyone. Is my slide is visible?
Yes.
Am I audible?
Yes, we can see your slide as well.
Yeah, thank you. So good morning, everyone. I'm Dr. Senthilkumar Palanisamy. I'm a NanoBio4Can MSCA COFUND Fellow at SUNUM, Istanbul. Thank you very much for everyone joining this webinar today. In this today's presentation, I'm going to present an ongoing research on multifunctional nanoparticles for retinoblastoma therapy. So in this presentation, I'm going to talk about the academic and professional background and key achievements of myself and background of the project, objectives of the project, followed by overall experimental design and workflow, and some scientific outcomes and the dissemination activities. So I am Dr. Senthilkumar Palanisamy. Basically, I'm from India. I completed my PhD in biotechnology in India. In my PhD, I have isolated a novel compound from marine algae. And I have also completed my post-graduation and undergraduation in Bharatiyar University, India. And also, I have more than 10 years of teaching and research experience in biotechnology departments. And there I have mentored undergraduate and postgraduate students. Also, I produced one PhD candidate in the year 2018, same nanotechnology field. And at present, I'm working as an NanoBio4Can MSCA postdoctoral fellow at SUNUM. Before we go into see about my project, I would like to highlight my key achievements. According to my research profile, I have so far published 40 research publications. Among the 40 research publications, I have 35 first author and corresponding author publications. And also, my publications received more than 1,000 citations with H-index 14 and ITAN index 18. And apart from the publications, I also published two books. One book is the Handbook on Practical Microbiology, and second one is Laboratory Manual on Plant Tissue Culture. I also have contributed to innovation and technology development through 4 patents have filed in our additional Indian patents related to nanomaterials based on food packaging. And also, I successfully completed the Indian government funded research project supported by the University Grants Commission India, entitled on green synthesis and characterization of gold nanoparticles from marine seaweeds for the treatment of type 2 diabetes. This project was focused on the developing environment-friendly nanomaterials for biomedical applications. Now let's talk about the project background. My project focuses on the retinoblastoma. Retinoblastoma is a rare aggressive form of eye cancer that develops in the retina. It almost exclusively affects babies and young children, typically under the age of 5. And this retinoblastoma is primarily caused by the mutation of RB1 gene. It is a tumor suppressor gene located in the chromosome number 13, and it is producing the tumor in the retina. And we have a lot of current treatments available with chemotherapy, laser therapy, cryotherapy, radiotherapy, and enucleation. That is the last one, the removal of eye. However, these treatments are producing some undesirable side effects, and also it is not that much good for the poor drug penetration, drug resistance, and also the vision loss. Hence, the recent scientific investigation have been reporting that the nanotechnology-based materials are very good for the treatment of retinoblastoma. Hence, the present investigation have been proposed to identify the potential nanomaterial-based therapy for the retinoblastoma. So, why we are using the nanomaterials? Basically, despite significant advances in cancer treatment, the conventional therapies often face other challenges such as non-specificity, resistance, limited efficiency, and also the invasive, limited personalization, and the cost. But on the other hand, we have a nano-enabled anti-cancer treatment that will help to uh, selectively deliver the nanotherapeutic agents to the tumor tissues and improving the drug accumulation at the target site, and while minimizing the damage of the healthy cells. It also helps bypass the resistance mechanisms and improves the effectiveness of anti-cancer drugs. And these nanoparticles improve the drug solubility, stability, and tumor penetration, leading to better treatment results. And also, these nanomaterials enable systems allow for a non-invasive or minimally invasive administration for great patient comfort. And also, the nano platforms can be delivered tailored to individual patients' need to for precise and personalized treatment. Finally, this nanomaterial treatment improve the targeting and reduce side effects. It can lower overall treatment cost and improve quality of life. So this is my overall formulation and the approaches of retinoblastoma. Here I'm using the multimodal approach. Multimodal, we have the core nanoparticles. We have two metal nanoparticles coated with the polymer. And also we'll be loading with drug and co-loaded with the peptide. We'll be creating the multimodal system. So this multimodal system addition, it will provides the nanoparticles to improve the targeted delivery to the tumor cells, while the drug provides anti-cancer activity and peptide supports the therapeutic responses. In addition, the nanosystem offers the imaging tracking capabilities, enabling the monitoring of treatment responses and nanoparticle distribution. By combining the nanoparticles, a polymer, a drug, peptide with a single platform, this multimodal approach aims to enhance therapeutic efficiency, reducing side effects, and improve overall treatment outcomes of retinoblastoma. And the research objective of my study is, the first objective is to develop multifunctional nanoparticles for targeted delivery to retinoblastoma cells. And second objective to enhance immune responses to support anti-tumor activity. Third one is assess cellular uptake and anti-cancer activity using in vitro studies. And the fourth objective is evaluate therapeutic efficacy and safety in suitable animal models. The final and the last objective is monitoring the treatment response and drug distribution using non-invasive imaging techniques. So this is the overall workflow of my research, and here I'm going to first objective of my study is to synthesize the nanoparticles using the chemical synthesis method. After successfully synthesize the nanoparticles, we'll be going with the characterization with different instrumentation using UV, FTIR, XRD, DLS, Zeta, SEM and TEM analysis. So after the synthesis and characterization of the nanoparticles, we'll be going to load the drugs along with the peptides, and also we'll be using the peptides. And the third experiment of my research is the in vitro evaluation of the studies. Here I'm going to use the retinoblastoma Y79 cells, and I'm going to assess the cell toxicity analysis, cellular uptake analysis, apoptosis and cell cycle analysis. And the final one is in vivo evaluation with mice. And the overall goal of the project is to develop and evaluate the targeted nanoparticle-based drug delivery system for enhanced anti-cancer efficacy and safety. So far, this is the outcome of my research. And so far, we have successfully synthesized the multifunctional nanoparticles. And also, we completed the characterization analysis with XRD, FTIR, DLS, Zeta, TEM, and EDS. And also, we have successfully coating with the polymer and performed the confirmed by the characterization system. And the drug loading and encapsulation efficiency studies are currently in progress, and also the peptide loading studies are being carried out. And in upcoming month, we are going to start the in vitro culture experiments, and also we have already received the cells and already subculture it, and we will be performing and evaluating the cellular uptake and cytotoxicity analysis. After successfully completing the in vitro analysis, we will be starting the in vivo animal model study to analyze or to assess the therapeutic efficiency and biocompatibility of the formulations. And this is the dissemination activities slide. And in addition to research activities, dissemination and professional development are important components of the NanoBio4Can program. As part of the activities, I delivered the presentation on retinoblastoma nanotechnology to school students and to helping the increasing the cancer research and biomedical innovation. The project was also promoted through social media and NanoBio4Can YouTube video presentation, allowing us to communicate our research to broader audience. Furthermore, I participated in technical training activities, including to visit TÜBİTAK MAM, where I gained the exposure to advanced research facilities and analytical techniques. I also attended a training and industry visit at Trust Life, which provided valuable insights into the biotechnology product development, commercialization, and industry academia collaboration. So these activities have helped strengthen scientific communication, networking, and professional skills while increasing the visibility and impact of the project. And finally, I would like to acknowledge the support provided by the European Union, TÜBİTAK, and the NanoBio4Can program. And also, I would like to thank my supervisor, Dr. Sibyl Chettinel. And thank you very much for your attention. I'll be happy to answer any questions discussed on this project further. Thank you.
Thank you, Dr. Senthil. Now we have Q&A session. You can just directly speak up, or you can use chat box.
Can I ask a question, Dr. Senthil?
Yes, please. Yeah, thank you.
Okay, so my question is, how would you confirm successful polymer coating on your hybrid nanoparticle system?
Yes, in my formulations, we have nanoparticles along with the polymer. It is a biocompatible polymer. And the successful polymer coating is confirmed by using the FTIR analysis, followed by the DLS Zeta. And also, we can confirm the for polymer coating with the TEM analysis. And so far, we have confirmed the successful coating with the FTIR analysis.
Thank you so much.
Thank you.
We have a question in chat box from Shoeb. Your formulation contains metal nanoparticles. If they remain in the body after administration, could they accumulate and cause toxicity?
Yes, this is a very good question actually. Uh, in my formulations, I have metal nanoparticles. Basically, we have a Fe3O4 magnetite nanoparticle along with gold. And this Fe3O4 nanoparticle is a biodegradable nanoparticles. Once it enters into the body, it can convert it into the Fe ions and it will be entered into the metabolism. And also it can be uh, involving the hemoglobin synthesis. And apart from the Fe3O4 nanoparticles, gold is inert and also it is not a biocompatible. But if we are using the very small quantity, and it might be eradicated with the liver or spleen with ALP or AS enzymes. Also, we are going to conduct some toxicity-based analysis with biodistributions and ocular toxicological analysis and other toxicity-based studies. If it is toxicity, we will be changing the formulations for the in vivo animal model studies.
Dr. Saif raised hand.
Yes.
Thank you very much for your last presentation. My question is regarding the execution. Is your project contain synthesis, in vitro validation, and then in vivo validation? So what do you think? Would you be able to complete the project within this time frame successfully?
Yeah. So far, I'm trying to complete the formulation studies. After that, also simultaneously, I'm performing the in vitro analysis. And as soon as completing my in vitro analysis, I'll be going to perform the in vivo animal model with the support of my supervisor. Most probably, I can complete.
Thank you.
Thank you.
Ankush has a question in chat box. Thank you, Senthil, for the presentation. My question is, how did you investigate the stability of the functionalized materials?
Yeah, in the stability of the colloidal solution, maybe, we are conducting the DLS analysis along with the zeta. And this DLS and zeta analysis gave us some better stability of our formulation.
Mina asked a question. Thank you, Senthil, for the presentation. My question is, how did you measure the loading of your peptide on the nanoformulations?
Yes, we have the peptide loading encapsulation efficiency by checking UV and HPLC analysis. The successful peptide loading efficiency after confirming will be going for the in vitro and in vivo analysis.
Melike asked a question. Thank you for the interesting presentation. I was wondering, from your experience, what has been the biggest challenge in achieving efficient targeted delivery to retinoblastoma?
Yes, in my formulation, I created the multimodal system. The ultimate aim of the nanoparticle system is to cure the retinoblastoma. And here we are, I'm using not only for the chemotherapy, but also I'm using the peptide for the immune booster. And also, these nanoparticles can help for the imaging analysis. So all these three systems can be helping to give the better results of the retinoblastoma treatment.
Thank you. Ankush wrote again, you mentioned about XRD. Does the structure of your functionalized materials intact to the WRT pristine material? Can you comment on structural stability?
Yes, we did the different XRD analysis. First of all, we did the Fe3O4 nanoparticle XRD, and there we have confirmed with the cubic planes of the nanomaterial, I mean nanoparticles. Second, separately we conducted the XRD analysis of the gold nanoparticles, and here also we confirmed, and also we used the hybrid, that is gold coated iron oxide nanoparticle with XRD, and that also gives the both Fe3O4 nanoparticle planes and also the gold planes. We have already confirmed with the chemist, and also the databases available for the confirming JCPD software for the confirming the XRD analysis.
Thank you, Dr. Senthil. Now we have, sorry, one more question. This is last one.
Okay.
Mustafa El Garib. Thank you so much for the presentation. There has been many articles in the literature about using nanoparticles for the treatment of retinoblastoma, as the articles from Stefan de Smet group in Belgium. Could you let us know how is your formulation stand out?
Yes, in previous literatures, I can gone through with retinoblastoma treatment. They have used only the chemotherapy, or they used only for the imaging studies. But here I'm using the multimodal approach. That is, I'm using the nanoparticle system along with the polymer coating, and also the drug along with the peptide. So all this will create with a multimodal system. These systems can help to not only the chemotherapy, and also we can check the immuno efficiency of the nanomaterial along with the imaging imaging of the retinoblastoma treatment. So all these things we can see in combinedly in with my research project.
Mina Masudi, this is last question. How do you measure the biocompatibility on the formulations?
So we are going to use the in vitro analysis and in vivo analysis. Both analysis can give the biocompatibility of my formulation.
Thank you, Dr. Senthil. Now we have Dr. Shoeb Anwar Ansari. This stage is yours.
Can you mute?
Yeah, please.
Turn off.
Yeah.
Marhaba. Thank you, Dr. Anam, for giving me the opportunity. Good morning, everyone. Now I'm sharing my screen. Now is visible?
Yes.
May I go?
Yes, please.
Okay. Good morning, everyone. My name is Shoeb Anwar Ansari, NanoBio4Can MSCA COFUND Fellow at SUNUM. Today, I will present my NanoBio4Can Fellowship project entitled The Overcoming the Blood-Brain Barrier, a novel in vitro approach using drug-loaded nanobubbles and a focus ultrasound for the glioblastoma. So let's start with the table of content. First, I will start with the professional and academic background, and then I will give the overall summary of my project, and then experimental design and some result of my outcome of the results and dissemination activity. Let's start with academic and professional background. I'm a pharmacist. I had a B.Pharm from the Pune University, India, and after completing my B.Pharm, I joined in the Bombay College of Pharmacy to do the Master of Pharmacy in a pharmaceutical advanced chemistry. Then I moved to Italy for the PhD in the complex system of life science from the University of Turin, one of the famous university in the Europe. And then after completing my PhD, I joined the same lab as a postdoctoral researcher. Then after completing my first postdoc, I moved to India and joined a pharmaceutical company named as the Dr. Reddy's Lab. Is one of the famous pharmaceutical company in India and also internationally. I joined as a formulation scientist where I learned how we can move the product from lab to the market. My role on this industry is like I have to product selection and ideation scientist. I have to work on the available dosage form and drug and how we can solve the unmet need of the patient. Then after this, I again moved to Italy and joined the University of Turin as a postdoctoral fellow. Then after completing my postdoctoral fellow, I joined the MSCA COFUND fellow here at Sabanci University. So far, I have published almost 23 articles, one book chapter, and 5 international patents. 2 is from the UK, 3 is from UK, and 2 is from the Germany. Along with, I also had the visiting researcher experience at different universities in Italy and other countries. Let's talk about the summary of my project. The main aim of this work is to improve the drug delivery for the glioblastoma. So in this work, we have the 3 work package. Basically, we call as the 3 main objective of my work. The first work package is like development and characterization of the drug-loaded nanobiological, and its optimization. In the optimization, we are performing the different characterization is like DLS, Zeta, SEM, TEM, and also we perform the in vitro toxicity study and an in vitro drug release study. The second objective of this work package is like the combination of GBM BBB on a chip model along with the focus ultrasound to investigate the focus ultrasound mediated opening of the blood-brain barrier. The third package is basically the combination of the package 1 and the package 2, where we are optimizing the drug delivery for the glioblastoma. So today I'm going to talk about my work package 1. It's like a development and characterization of drug-loaded nanobubble. Let's start with the glioblastoma, global disease burden. And the glioblastoma is the most aggressive brain tumor. It's the glioma, type 4 glioma. So beside this, the current therapeutic approach is still the medium survival rate of this glioblastoma is 14 to 18 months. Along with this, only less than 5% people can survive up to the 5 years range. And if you are seeing the statistical data, this glioblastoma cases, the number of the cases is increases if you see in the US data, 10,000 cases per year. And if you are considering the global data, 250,000 cases per year. So most of the cases are in the developing nature because of the environmental factor and stress things. So be careful, don't take too much stress in the life and be happy. And the most saddest part of this glioblastoma treatment is the reoccurrence. 90% of the patients facing the reoccurrence within the 6 to 9 months. And after the treatment challenges and unmet need, the blood-brain barrier limits the chemotherapeutic access. As we all know, the human brain is the central organ of the body, and it is protected by the multiple barriers, such as the blood-brain barrier and the cerebrospinal fluid barriers. So here we are coming to solve this problem by combining the focus ultrasound and the nanotechnology to overcome this blood-brain barrier and reach to the glioblastoma. So what is the nanobubble? Nanobubble are the gas-filled nanostructure. Basically, it has the core and the shell. Inside the core of the nanobubble, it has the gas, perfluorocarbon, and oxygen, whatever you want, you can add inside the nanobubble. And the shell of the nanobubble is like a lipid, protein, any drug, polymers you can use. Nanobubble is the most famous for theranostic approach. It can be used for the drug delivery, also for the diagnostic imaging. Let's talk about the focus ultrasound. The focus ultrasound is a non-invasive technique that can transiently and reversibly open the blood-brain barrier. And This focus mechanism behind the focus ultrasound is like that the focus ultrasound waves precisely focus on a small targeted region within the brain, and these waves increase the acoustic pressure locally and without affecting the other tissues membrane. How it's work when we are injecting the nanobubble systematically, and when focus ultrasound come in contact with the nanobubble is cause the cavitation and oscillation technique. As you see in the video, because of this oscillation, it exerts the mechanical force, or because of this mechanical force, the tight junction of the blood-brain barrier open reversibly for 4 to 24 hours. How nanobubble can form? It's a multi-step procedure. First, we prepare the core and then shell. We synthesize the both positively charged and negatively charged nanobubble. In my nanobubble form, we loaded the drug inside the core of the nanobubble, here inside the core of the nanobubble, and then there is a layer of the lipid and the shell. Physicochemical characterization, as you see the DLS data, it's almost the nanobubble is the less than 200 and nanometer is around in the range of the 180 to 200 nanometer. Also, the PDI is the less than 0.3. That means it's the monodisperse. With the help of scanning electron microscopy, we confirm the spherical shape of the nanobubble and its size, and also there is no aggregation is formulation. The encapsulation efficiency of the nanobubble is around 70 to 74.5%, and the loading capacity is around 15%. We performed the in vitro release study in the both physiological condition and the acidic condition. As we know, in the acidic condition, the release of the drug is increased as we want this kind of the conditions. In the acidic condition, we need a more release of the drug. So it's a preferentially an optimized drug formulation, which we increase the drug release at the physiological condition. We perform this release study until 144 hours. That means the 6 hours. And also we test this nanobubble, blank nanobubble in the MTT. All the nanobubble are safe. Along with this dissemination, I am the mentor of the one undergraduate student, and the 2 more pure program student of the Sabanji University will come the next week. Along with this, I performed the many training in TÜBİTAK MAM, IBG, and KUTAM. I also gave the one public talk to the high school student, and 2 months before, we also visited the Trust Life, the pharmaceutical company, to gain the knowledge how we can move to the market, and also attend the last year the conference of EACR. I would like to thanks to MSCA, European Union, and TÜBİTAK for giving me this funding, and I would like to thanks the entire team of NanoBio4Can, the SUNUM, IBG, TÜBİTAK, and KUTAM for providing the training and giving the healthy environment and all these things. Last but not least, I would like to thanks to my lab, Mustafa Glue Lab, and also my mentor, Dr. Noor Mustafa. She's such a nice person. She's helping a lot to solve all this problem. Do you have any question? Let's go.
Thank you, Dr. Shoeb. Now we have Q&A session. From
I have one
Janer,
question.
you have a question.
Can I go first?
I think Janer wrote, Kashif. After Janer question, yes. How do you ensure that the focused ultrasound nanobubble combination increases BBB permeability without causing toxicity and permanent damage to the blood-brain barrier?
We are using the microfluidic BBB on a chip model, and then on this model we will analyze and optimize the focus ultrasound, its pulse rate and its frequency, what frequency we have to use. We are going to optimize this to solve this toxicity. Also for the toxicity, we are targeting at a particular area. not whole the brain. So it's a targeted drug delivery system. So there is other than this glioblastoma cell, there is no damage.
Yes, Kashif.
Okay, Dr. Shoeb, can you go to the slide number 8?
Yeah.
Yeah, so in this slide, you mentioned when you apply the focus ultrasound, it would cause the blood-brain barrier to open temporarily. So I would argue that temporary opening would also allow the other harmful substances like bacteria, viruses to enter the blood-brain barrier. So how do you make it selective only for nanobubbles?
Thank you, Kasif, for this question. Basically, we will performing this experiment in a sterile condition. Also, our nanobubble size, as you see the DLS data and in SEM data, the size of nanobubble is very small, is less than 200 nanometer. So if you are applying the focus ultrasound, it open the blood-brain barrier according to this, like this size, and the capillary size of the brain is very small. If the bacteria and other thing, if you see the size of the bacteria pathogens is very high, but it's a good thing for the toxicity purpose and also the patient safety concern. We will take a look on this. Also, if you are seeing this is a non-invasive technique, but invasive technique, they are performing under hospital operation theater, they are opening the brain. So it's also the chance so that we are going to the sterile condition and also the like this.
Yeah, thank you.
Dr. Saif,
Yeah, thank you, Dr. Shoeb, for your nice presentation. My question is regarding its delivery and release of drugs. What would be the factor which release the drug from the nanobubble at the site of specific location? Is it pH, temperature, or ultrasonic signal, or what would be the key things?
Okay, this is the multimodal. Nanobubble is famous for the drug delivery with the help of ultrasound. So here we are also using the focus ultrasound. Also, this drug has the pH response delivery. As you see at 5.5 pH, it increased the release of the drug, in vitro release of the drug. So it will reach at a tumor microenvironment, the pH is like a 5.5. So if it reached to 5.5 pH in that particular tumor, so it increased the release of this drug.
we have a question in chat box. Mustafa El Garib, thanks for the presentation. Could you give us more info on frequency and duration you used? Is it doable with US machine or any collateral damage exposed to adjacent tissues?
Sorry, I couldn't get what exactly.
On the chat box,
Okay.
Mustafa El Garib.
Or I could say it actually, like if you hear me. So actually, like the question is actually about like could the gas? Okay.
You can speak up, Mustafa.
Yeah, I'm asking actually about the frequency that you use for the ultrasound and the duration that you actually like expose your tissues to. This is one thing. This is actually like doable with the ultrasound machine, like the normal ultrasound machine, like the people are using actually like in the in the clinic. And also something asked about the collateral damage. Like this should be like focused. This is something like in the place. But maybe like we have like a leakage, like if you load your nanoparticle with the, for example, a cytotoxic component, so it would be like released upon the this focused ultrasound to like adjacent tissues. It will be like damaging to the normal tissues or not? Thank you.
So we are using the low intensity focus ultrasound. So it will not damage to the other part of the brain. So we are directly focusing on the particular tumor site, and then we are directly release the drug on that site. There are two types of the ultrasound. It's high intensity, low intensity. Also, the application time is we are using very short time. Any other?
Yes, we have question from Senthil. There are several nanocarriers available for drug delivery to the brain, including liposomes, polymerics nanoparticles, solid lipid nanoparticles. Why did you specifically choose nanobubbles for your project?
Okay, so nanobubble is helpful for the focus ultrasound because it's compressed after applying the nanobubble has the gas inside the core. If you are applying the focus ultrasound, it compresses and like extraction and relaxation. And other nanosystem, like if you are saying the polymeric nanoparticles or liposome and other thing, they have the core has the liquid inside. So it's not to work with the focus ultrasound if you are using. The nanobubble has the gas. So because of this gas, it can compress.
Any questions? Fazel, please. You're muted.
Hello.
Hey.
Thank you very much for your nice presentation. I have one question. Do we have the enhanced permeability and retention effect in the tumor tissues in brain? And how does it align with your research and its effect on blood-brain barrier?
Okay. Now we are going to perform these experiments. So we are testing this blood-brain barrier permeability, and also we are exploring the TR value. And we are using the fluorescence agent to see how its blood-brain barrier permeability through the BBB.
Do you have the model for this?
My lab is working on the BBB models. So
only on BBB models or any kind of vascular permeation models?
till now, I can say that we had a BBB
Because
because
I'm
my
asking this.
supervisor is expert in this blood-brain barrier.
I'm asking this because it's also a question for myself, and I'm wondering if I can get help from your group for part of my work.
You can.
That's why I'm asking this.
Okay, you can contact us with Noor Hoja, and also you can contact to me. We will discuss this in detail. What exactly you want to.
Fazal, we will be happy to help you.
Noor Hoja is here.
Thank you. Thank you. Then I will email you later. And one question is the measurement of echogenicity. How do you do it?
Measurement of?
Echogenicity.
What exactly?
No, when we pass the ultrasound waves through the body, according to its speed absorption or passing through it, or the speed of passage through different tissues. This is also valid about any kind of material. There is a specific pattern of getting the waves back into a detector or absorbance of the ultrasound waves by the tissue. Do you have any kind of measurement? Have you done any kind of measurement for that or not?
Not yet, but focus ultrasound is a well-established method for the BBB opening. Is almost, I think it's 21 or 23 clinical trial is going on this focus ultrasound for the blood-brain barrier and other things.
I see. Thank you.
Thank you. Any other?
Thank you for your presentations. Thank you, Dr. Saif Ullah Afridi, Dr. Senthilkumar Palanisamy, and Dr. Shoeb Anwar Ansari for your presentation. And thank you all participants for your participations.
Thank you.
Thank you.
Thank you,
Goodbye
Nokta.
and good luck.